This week we will explore the biological models of substance misuse. We will also talk about psychopharmacology principles. You will be able to explain evidence concerning the genetic basis of substance misuse. You will be able to describe the role played by different brain regions. Also, you'll be able to describe the roles of neurotransmitters. You will know why age of substance use initiation is crucial for disorder outcomes. You will learn about the role of homeostasis. And finally, you will be able to describe the basic principles of pharmacokinetics and psychopharmacology, drug half-life, synergism, agonists, and antagonists, and how this might relate to medication for assisting in treatment of substance use disorder. First of all, let's review the key terms used in this module. On this slide you can see a DNA, or deoxyribonucleic acid, is a molecule composed of two polynucleotide chains that coil around each other to form a double helix, carrying genetic instructions for the development, functioning, growth and reproduction of all known organisms, and many viruses. An allele. An allele is a variant form of a given gene, meaning it is one of two or more versions of a known mutation at the same place on a chromosome. What is a gene? A gene is the basic physical and functional unit of heredity. Genes are made up of DNA. Some genes act as instructions to make molecules called proteins. However, many genes do not code for proteins. In humans, genes vary in size from a few hundred DNA bases to more than 2 million bases. The Human Genome Project estimated that humans have between 20,000 and 25,000 genes. What is a chromosome? In the nucleus of each cell, the DNA molecule is packaged into thread-like structures called chromosomes. Each chromosome is made up of DNA tightly coiled many times around proteins called histones that support its structure. A genome is an organism's complete set of DNA, including all of its genes. Each genome contains all of the information needed to build and maintain that organism. In humans, a copy of the entire genome, more than 3 billion DNA base pairs, is contained in all cells that have a nucleus. Genotype, very simply, is the version of a DNA sequence that an individual has. There's a large amount of DNA that we all have in common, of course. That's why we're all humans, but there's also a large amount of variation in sequence among individuals. And those specific differences in sequence, when usually applied to an individual gene, are called a genotype. These days, with the ability to test for many different sequence differences between individuals, genotype has taken on a connotation which frequently refers to a difference in sequence in a specific place in a specific gene. When used in that way, it's usually related to another term, called phenotype, which is the change in sequence, to which the genotype refers. It is frequently, not always, but is frequently related to a change in an external trait. Something that's observable, like height, hair color, or occurrence of disease. And so in that case, we talk about a genotype-phenotype correlation. Then what we're talking about is, well, here's a change in DNA sequence. Why is it important? It's important because it leads to an observable change in a trait in a person. And that change in trait can be positive, it can be negative, or it could just be a difference. What is heritability? Heritability is a measure of how well differences in people's genes account for differences in their traits. Traits can include characteristics such as height, eye color, and intelligence, as well as disorders like substance use disorder. In scientific terms, heritability describes how much of the variation in a given trait can be attributed to genetic variation. An estimate of the heritability of a trait can change over time as circumstances change. Heritability estimates range from 0 to 1. A heritability close to 0 indicates that almost all of the variability in a trait among people is due to environmental factors, with very little influence from genetic differences. Characteristics such as religion, language spoken, and political preference have a heritability of 0, because they are not under genetic control. A heritability close to 1 indicates that almost all of the variability in a trait comes from genetic differences, with very little contribution from environmental factors. Now let's talk about genetic influences. Individuals with genetically close relatives experiencing a substance use disorder involving opioids, cocaine, cannabis, or alcohol have up to an 8 times higher risk of developing a substance use disorder themselves. However, there is no one specific addiction gene that applies to all of the different types of substances. Some of the genes involved are very specific to certain substances. What may pull for an alcohol use disorder may not be pulling for a problem with cocaine, for example. Pedigree charts, or genograms, are often used to see familial patterns of substance use. The figure you see on this slide shows a family's pedigree for alcohol use disorder. Family members with alcohol use disorder are shown in dark red color. Adults with alcohol misuse are shown in light red. You can see 3 generations. The youngest generation are still too young to know about. Another source of evidence supporting the theory that alcoholism has a genetic basis comes from twin studies. The logic behind twin studies is to look at the degree of phenotypic similarity on some trait, like observable drinking behaviors, called concordance, between identical versus fraternal twins. If the degree of concordance is considerably greater among identical twins, this constitutes strong evidence for a genetic influence. Adoption studies are based on comparing the phenotypic outcome of children raised by their biological parents with children raised by adoptive parents, when the biological parents exhibit the phenotype of interest. In this case, children who are raised by the biological parents with an alcohol use disorder are compared with children raised by adoptive parents who do not experience alcohol use disorder. Evidence suggests that among children whose biological father experienced an alcohol use disorder, being raised in an adoptive family was moderately but not entirely protective. In other words, there is still a considerable genetic risk, about 50 to 60%. The human genome is a person's complete set of DNA, represented in virtually every cell of the body. The Human Genome Project, completed in 2003, resulted in a generic map of the approximately 20,000 to 25,000 genes in the human genome. Genome-wise association studies, GWAS, approached the study of substance misuse and SUD in a unique manner, searching for common variants in allele frequency across the entire genome, and then determining what phenotypic differences were associated with those variants. What is known about the genetics of substance misuse? First, there is clearly a genetic influence on the development of alcohol use disorders. Genetics are also likely to influence the development of disorder related to the use of other psychoactive substances. For example, initiating tobacco use during adolescence was anywhere between 35% to 80% heritable. Across different studies, regular tobacco use was between 40% to 50% heritable, and regular alcohol use was about 40% heritable. Second, evidence points to multiple genes contributing to substance use disorder, even to a single type of substance use disorder, such as alcohol use disorder. Third, researchers also found that genes involved in controlling the processes of alcohol metabolism in the human body demonstrate a potential for preventing alcohol use from becoming an alcohol use disorder. This happens because persons with such gene expression tend to have a highly unpleasant physiological response to alcohol use. Fourth, genetic influence is not simply occurring at the level of specific chromosome sites, but also in chromosomal regions. These regions are areas where multiple chromosomal sites cluster. In addition, genetic influence shows in various polygenetic combinations, when multiple genes interact with each other. Fifth, there seems to be some shared commonality in genetic vulnerability to nicotine, alcohol, and cannabis dependence, at least among men. There are also specific genetic factors for alcohol, tobacco, marijuana, and cocaine. One common underlying genetic factor may be the presence of genetics linked to depression. For some individuals, depression and SUD have common genetic influences. Sixth, the heritability of alcohol use disorder has been stronger for men. In other words, environmental factors explained a greater proportion of alcohol use disorder among women. However, this gender-based differentiation in alcohol or nicotine use has become very weak in the recent studies. Neurobiology investigators are developing increasingly complex, detailed, functional maps of the various regions of the brain involved in substance use, misuse, and substance use disorder. These maps show how the brain's powerful pain, pleasure, reward, and memory systems interact in the process of substance use, becoming a substance use disorder, and how psychological learning principles operate at a neurobiological level. There are certain brain regions identified as having a significant role in the development of substance use disorder. Also, learning about the neurochemistry actions of specific substances also helps us understand the actions of different substances on the brain behavior link. We will look at neurotransmitters and their role in the experience of substance use and misuse. Current technologies, such as functional magnetic resonance imaging, help develop our understanding of how different areas of the brain are involved in specific experiences or behaviors, and how exposure to different events or substances might affect specific brain areas and functions. There are certain brain regions identified as having a significant role in the development of substance use disorder. Second, the brain behavior link is influenced by, and influences, the autonomic nervous system, which controls many bodily functions, such as heart and breathing rate, blood pressure, and others. Many psychoactive substances not only affect the mind, they also affect other organs and systems, including the autonomic nervous system. Third, the limbic system helps regulate basic drives, emotions, arousal, and attentiveness. As such, it helps coordinate the neurobiological experience of stress and the reward system, triggered by exposure to drugs. The amygdala, nucleus accumbens, and hippocampus are components of the limbic system, associated with substance misuse. Amygdala. The amygdala plays a central role in emotional responses to internal and external stimuli, pleasure, fear, anxiety, and anger included. It is central to survival, as it manages the fight or flight response to perceived threats in the environment, which in turn is related to the experience of stress. The amygdala is also responsible for the emotional content of our memories, determining not only which experiences, related to pain and pleasure, become encoded into memory, but also the emotional values attached to the formation of new memories. This area is one target of anti-anxiety medications, but is also influenced by the actions of various substances that might be misused. Hippocampus. The hippocampus is involved in memory, as well, particularly memories related to traumatic events and learned responses to environmental cues. This becomes an important factor in the experience of cravings, triggered by environmental cues, as well as the relationship between trauma and substance misuse. Nucleus accumbens. The nucleus accumbens is part of what is called the mesolimbic dopamine system. It is highly involved in positive reinforcement, leading to a person anticipating reward, with repetition of the previously positively reinforced behavior. Thus, if a substance increases the release of dopamine in this area, the person comes to anticipate positive reinforcement again, with future use. The amount of dopamine increase can far exceed what natural behaviors trigger, eating or sex, for example, and the amount of dopamine directly relates to the degree of pleasure experienced. Thus, a person may come to preferentially engage in substance use over naturally rewarding behaviors, like eating or sex. Prefrontal cortex. The prefrontal cortex is linked to the amygdala. They communicate directly. This is a thinking part of the brain, where functions like cognition, comprehension, concentration, reasoning, planning, and initiating goal-directed behavior takes place. The area is responsible for a person's intentional responses to the experiences the amygdala sends forward. For example, the conscious decision to initially engage in substance use. When prefrontal cortex is damaged by substance use, the person may not be able to mediate the responses to amygdala's push to action. Such person may be more impulsive and have a higher risk of relapse. Studies using functional magnetic resonance imaging clearly show that heavy, repeated, chronic substance misuse leads to changes in brain functioning. These changes remain long time after substance abuse stops. Over time, however, the brain begins to recover. The image on the left shows a brain of a person who has never engaged in cocaine use. The other two scans represent a person who has a history of cocaine use disorder, 1 month and 4 months after use has ceased. As you can see in these images, there is some improvement at 4 months post-use, but the function has not returned to normal. The density of dopamine receptors, which are shown as red dots on the left image, has not been restored. A great deal of attention to the developmental effects of exposure to alcohol and other drugs has been directed to 2 life periods. These periods are prenatal child development and adolescence, or emerging adulthood. These 2 developmental periods have an important commonality. During these periods, the brain is naturally undergoing rapid developmental growth and change. Thus, introducing substances that affect the brain can have more pronounced, amplified, and pervasive long-term effects. That alcohol exposure during fetal development can cause permanent damage to the brain and other organs has long been recognized, and fetal alcohol syndrome was clearly identified as a possible outcome during the 1970s. Subsequently, a fetal alcohol spectrum disorders have been described. It includes the fetal alcohol syndrome, as well as alcohol-related neurodevelopmental disorder and alcohol-related birth defects. The effects of prenatal exposure to other substances is less well understood. Neonatal abstinence syndrome is a known consequence, experienced by many infants, prenatally exposed to opiates. Shortly before and during puberty, the human brain begins to undergo dramatic remodeling changes. On one hand, a great deal of neuron pruning takes place, trimming out a great many, underused, or unused, connections between neurons. On the other hand, myelination of existing neurons enhances connections between neurons that remain linked. These two processes make the brain more efficient, better integrated, and capable of higher order functioning. However, while the emotional functioning is similar to that of adults, the cognitive functioning is as yet underdeveloped in terms of decision making, inhibitory control, planning, and working memory. The brain revision process normally tapers off at about ages 20 to 25. This image shows how the concentration of gray matter shifts from age 5 to 20. The shift from yellow to blue in these images. Thus, the brain is quite sensitive to developmental consequences of exposure to psychoactive and psychotropic substances up until age 25. The use of alcohol or other substances during these years can have profound, lasting effects on the still developing brain. Chances of developing severe substance use disorders is higher among individuals whose substance use began before age 15 years. The biggest reduction in risk, with deferred age of onset, occurs when first use is postponed beyond age 15. The neurochemistry of substance use operates largely at 2 points. The first concerns the glial cells and how much energy they can provide to neurons. The loss of glial cells, or impeding their ability to provide energy, has a negative impact on neuronal activity. The second concerns the ways that neurons communicate. Neurons physically pass neurotransmitters, which naturally occur in the brain, between each other, as their mechanism for communication. On this picture you can see the neurons and their communication. The sending neuron is called the presynaptic neuron, while the receiving neuron is the postsynaptic neuron. The presynaptic, first neuron, releases neurotransmitter molecules, stored in the vesicles, into the synapse, between it and the postsynaptic, next neuron. The postsynaptic neuron receives the neurotransmitter chemical, if it has the right neurotransmitter receptors, kind of like a lock and key system. On this picture, you can see that neurotransmitters need the right receptors in order to dock and influence the postsynaptic neuron. If the right receptors are available, the neurotransmitter delivers the message. But if the right receptors are not available, the neurotransmitter has no effect and just sits in the synapse. If the message is received by the postsynaptic neuron, it can now pass the message along to the next neurons in line. In the meantime, transporters retrieve and return the used neurotransmitter molecules back into the presynaptic neuron's vesicles, in preparation for sending a future message. If the postsynaptic neuron's receptors are already filled, then the sent message will not be received, the neurotransmitters are blocked. This is how some drugs work. They occupy the receptor sites, thereby blocking messages between neurons. Other drugs work to reduce or increase receptor site sensitivity to the neurotransmitters. Still others work to influence the amount of neurotransmitter released into the synapse, or affect the transporter's work in returning the neurotransmitter molecules to the vesicles. Different types of neurotransmitters have different impacts. For example, some play a more excitatory role, while others play a more inhibitory role. Excitatory neurotransmitters increase the likelihood that the receiving postsynaptic neuron will be triggered into activity. Inhibitory neurotransmitters suppress this kind of activity. Most types of neurotransmitter are either excitatory or inhibitory. A few can be either, for example, dopamine. Different types of neurotransmitters are more concentrated in specific brain regions. While they may be distributed throughout the brain, they are not evenly distributed. This is why different substances trigger certain brain regions more than others. Their effects are produced through their influence on the neurotransmitter communication processes, and those neurotransmitters are more concentrated in certain regions. Neurotransmitters occur naturally in the body and have health-related functions. Neurotransmitter release is triggered by many natural behaviors, not just by alcohol and other substances. For example, dopamine release is involved in the natural reward systems associated with food, sex, humor, pair bonding, listening to music, and video games. A number of neurotransmitters are triggered by substances and begin to play a crucial role in substance abuse. Various functions adjust to conditions in order to maintain overall balance. This balance is called homeostasis. You may recall from Module 2 that we defined acquired tolerance as a person requiring higher doses of a substance to achieve the same effects. Let's consider what is happening at a neurochemical level. When a person uses a great deal of alcohol, often over time, the brain begins to adapt to the presence of the alcohol and its effect on GABA. GABA is responsible for feeling calm. In attempting to reacquire a state of homeostasis, the brain boosts its arousal systems, glutamate, to offset the overly inhibitory impact of the extra GABA triggered by the alcohol. This is called upregulation of the glutamate system. The brain additionally activates the system that produces glutamate. In addition, the brain may begin to control the amount of GABA through downregulation of the GABA system, suppressing the system that produces GABA. This means that, in order to experience the same effects at the same level, a person needs to take even more alcohol to boost the GABA even more. Now, let's look at the other side of the coin. What happens when the brain is no longer exposed to substances to which it has grown accustomed? Remember that the brain has adapted to the chronic presence of the substance, alcohol in our example, by downregulating GABA and upregulating glutamate systems. Withdrawing the alcohol means that the GABA and glutamate are going to be out of balance for a while. The withdrawal of substances can result in the experience of withdrawal symptoms, an experience that may be intense, even potentially deadly, and prolonged. A decrease in dopamine or serotonin contributes to the experience of dysphoria and anhedonia. Dysphoria is the experience of a profound sense of unease, unhappiness, and general dissatisfaction, often associated with major depression and anxiety. Removing substances that stimulated dopamine or serotonin activity can have these effects. A decrease in GABA contributes to the experience of anxiety, even panic attacks, due to the resulting nervous system hyperactivity. Why does this matter? These negative emotional and psychological states make it difficult to sustain motivation to avoid using alcohol or other substances, and contribute to the pressure a person might feel to relapse into using again. In this chapter, we introduce some basic principles of pharmacokinetics and psychopharmacology. Pharmacokinetics is the study of how drugs are distributed and metabolized, broken down, in the body. We are talking here about the patterns by which different substances are absorbed, metabolized, and excreted. It helps explaining overdose and differences in how quickly different substances begin to have an effect, or how long the effects might last. Psychopharmacology is concerned with how different drugs have their effect on the brain. We will talk about how different drugs might influence the actions of different neurotransmitters, as agonists, antagonists, and synergistic effects. These actions have implications for pharmacotherapy, meaning the medications that can be used to treat substance use disorders. Half-life. The duration of a drug's effect is measured in terms of its pharmacological half-life, which describes the relationship between the active dose circulating in the body, its concentration, and the variable of time. From the time of administration, different drugs take a different amount of time to reach peak level. Then, as a drug is metabolized, there comes a point in time when its circulating concentration is half of what it was at its peak level. The time that it takes to achieve this point is called the distribution half-life. The first half-life is the point when 50% of the drug is gone. This means 50% remains. The second half-life is where 50% of what remained after the first half-life is gone. In other words, another 25% is gone, half of 50. Together, this means the original 75% of the peak level is gone, or only 25% remains. The third half-life is where 50% of what remained after the second half-life is gone. Only 12.5% remains now. And so on, until virtually none remains. This curve might help you visualize the relationship of half-lives and time for a hypothetical situation. The principles behind the curve are the same for every drug. It is the length of time for each half-life that differs. It could be minutes, for example, for some inhalants, hours, or even days. This also affects how long after using a substance it can still be detected in drug tests. Alcohol can be detected for 7 to 12 hours or more after drinking in a urine test. Marijuana can be detected in urine for about 3 days for some who uses it occasionally, and for more than a month after last use by a person who uses it multiple times a day. Opioid detection is possible in urine tests for 2 to 4 days, for the most common forms, and this is about the range for detecting cocaine metabolites in urine as well. This curve shows the relationship of half-lives to dose effect. The distance between peak dose and overdose differs by drug. In some cases, there is very little wiggle room, making it very easy to end up with an overdose, the red line in the curve. This is true, for example, of barbiturates and benzodiazepines. The difference between therapeutic and overdose range can be quite narrow. As the dose of a drug increases, so do the risks of side effects, even below the overdose level. In this hypothetical example, a person might need to take more of the drug at the point where 50% remains, first half-life, in order to maintain an effective dose. But it is going to be important to avoid a peak dose that takes that person into the overdose range. What is known about half-lives, effective dose range, and overdose range is based on averages across individuals. It may differ for a single individual and by various conditions, including the person's overall health and presence of other drugs. Individuals differ somewhat in how they metabolize drugs. The breakdown process for many substances is not as simple as, there and gone. In many cases, the process of metabolizing a drug or other substances happens in a sequence of steps, and the intermediary products may exert effects themselves. For example, alcohol, ethanol, is first metabolized, broken down, into another chemical called acetaldehyde. Acetaldehyde is toxic and thought to be responsible for many of the hangover symptoms associated with alcohol consumption, as well as with the increased risks for cancers. Fortunately, acetaldehyde does not stick around very long as it is metabolized into a less toxic chemical, acetate. Acetate is then metabolized into carbon dioxide and water. The enzymes responsible for the metabolism of ethanol are under genetic control. This contributes to the observed phenotypic differences in individuals' responses to drinking that we previously explored in discussing genetics. Agonists, antagonists, and synergism. An agonist activates specific types of receptor sites in the brain or elsewhere in the body, causing a specific effect. For example, THC is a chemical in cannabis, marijuana, that activates the naturally occurring cannabinoid receptors in the brain. Methadone can help in the treatment of opioid use disorder. It acts enough like the opioid to help without the added risks and potential harms of using the original substance. Antagonists. Antagonists mostly work by blocking receptor sites in the brain so that a drug cannot trigger its expected response. For example, naloxone is used as an emergency first response to heroin or opioid overdose. This potentially life-saving medication blocks the effects of heroin or other opioids. Synergism. Certain substances, when combined, create a stronger or more prolonged response than either could alone. This is called synergism. For example, the combination of alcohol and barbiturates amplify the central nervous system's depressant effect, which is why it is easy to overdose on this combination. This concludes the lecture on the Module 3. Read the module carefully, as it contains many important details that may have not been included in this lecture. This module is relatively long, but remember you have 2 weeks to master its content.